Can you see my slide there? Should be there now.
Yes, we can see.
Okay,
Very
excellent.
clearly.
Thank you. So yes, hello everyone. My name is Helen Alexander. I am a Dermatology Clinical Research Fellow from St John's in London. And I'd like to thank very much Sandipan and the organisers for giving me the chance to speak today on the skin microbiome in atopic dermatitis. So I'll first of all give a bit of background and a bit of context and speak a bit about the healthy skin microbiome, and then move on to what we know about the skin microbiome in atopic dermatitis, and then speak a bit about how the skin microbiome differs in different geographical regions around the world and in different ethnic groups. So firstly, just to make the point that the reason why research into the skin microbiome and the microbiome in general has really increased over the last 10 to 20 years is because of advances in our methodologies for identifying skin microbes. So previously, the only way that we would be able to detect which microbes were present on the skin was through culturing and growing these species. And in the case of bacteria like Staph aureus, shown here, this is quite straightforward. But for the vast majority of bacteria, and particularly viruses and fungi that live on the skin, this is pretty much impossible. And so up until more recently, we were pretty much unable to identify what species were actually present on the skin. And so now we use microbiome sequencing to allow us to detect the species present on the skin. And this method starts off in a very similar way with a skin swab that's rubbed on the skin, and this picks up a mixed community of microbial species. And then the DNA is extracted from these and sequenced in the lab. And these sequences are then aligned to reference databases, which allows us to identify which species are present and then their different proportions of these species in our samples. And we can then compare these proportions in, for example, AD patients with healthy controls and identify differences in the skin microbiome in these diseases. And then because we sequence the entire genomes of these microbes, we can identify microbial genes that are present. And this then gives us insights into the potential function of the skin microbiome. So in terms of the healthy skin microbiome, we know that the skin provides a very diverse environment for a whole range of microorganisms to live. And any factors which alter the state of the skin will alter the environment of these microorganisms. And this will then lead to changes in the microbiome. So, for example, thinking about age, at the time of puberty, the environment of the skin changes a lot with an increase in sebum in the skin. And this then changes the skin microbiome to favor lipophilic bacteria, such as Corynebacterium and Propionibacterium, which are seen to significantly increase at the stages of puberty. And we also know from studies of the healthy skin microbiome that skin site is very important and that different regions of the skin are colonized by different microbes. So you can see here that these skin sites, such as the forehead and the chest and the upper back, the more sebaceous sites, these tend to have higher abundances of these blue organisms, which are the Corynebacterium and the Propionibacterium, the lipophilic bacterium that I mentioned, that are higher in these sebaceous sites. And this is then in contrast to more dry skin sites like the volar forearm, which has a more diverse microbiome. And then again, in contrast to more moist sites, such as the antecubital fossa, which favors staphylococci. And as you know, the common skin diseases like psoriasis and atopic dermatitis and acne are also all very site-specific. And this is part of the reason why it's been suggested that the microbiome may play a role in the pathogenesis of these diseases, because it also is very site-specific. And this study just confirmed that by showing that different skin sites show differences in the skin microbiome that are greater than differences between different individuals. So here you have 4 healthy individuals who had their microbiome sampled at 4 different skin sites: the antecubital fossa, the back, the nostrils, and the heel. And you can see that across the 4 individuals, the skin microbiome was very similar at each individual site. But when you look between skin sites, even within the same individual, the microbiome is very different. So skin site is an important factor in the skin microbiome. And we know that there is likely to be a number of important interactions between the skin microbiome and the host and the cells of the skin. So these microorganisms are living, they produce a number of factors such as toxins and metabolites, and all of these will interact with the keratinocytes and with immune cells in the skin to potentially induce changes that may be relevant in skin disease. So moving on to the skin microbiome in atopic dermatitis. So we know that Staph aureus is a very important organism in the disease. It's the most common organism to cause infection in atopic dermatitis, shown by these clinical photographs here. Myself and Sandipan were involved in this paper, which was really reviewing the most recent evidence on the role of bacterial skin infections in atopic dermatitis as part of the International Eczema Council. And so this paper really goes into this in quite a lot of detail. And we've also known for a long time that most AD subjects are colonized by Staph aureus, even when they don't have these overt signs of clinical infection, compared with only 5% of healthy individuals. And we know that topical and systemic antimicrobial treatments are very important in the treatment of AD and one of the most commonly prescribed treatments for the disease. And studies for a long time have shown that there is some benefit in treating with antimicrobials in addition to corticosteroids in atopic dermatitis, even when there isn't signs of clinical infection. However, other studies have not shown this, and in a recent Cochrane review, it was reported that there is not sufficient evidence to show that anti-staphylococcal treatments are clinically helpful in people with eczema which isn't clinically infected. So I would say that the question of what is the role of Staph aureus in atopic dermatitis still remains. Is it really a causative factor in the pathogenesis of the disease? Does it just exacerbate pre-existing AD, or is it simply a bystander of the inflamed skin environment in atopic dermatitis? And more recent skin microbiome studies have moved us forward towards answering this question. So this study confirmed that what we knew, which was that Staph aureus is increased in atopic dermatitis, that it increases during flares of the disease, and after these flares are treated, the Staph aureus abundance reduces again. And they also showed that the abundance of Staph aureus correlates with the severity of the disease measured by objective SCORAD. And also in this study, they showed that this increase in Staph aureus during flares of AD is usually a single strain of the bacterium. So we know that there's over 200 different strains of Staph aureus, and these have quite high variability in their genomes and in their functions. And it's suggested that during flares of AD, it's a single strain that really increases. So suggesting that it may be strain properties of Staph aureus that's important in atopic dermatitis. It's also known that the overall skin microbiome diversity is reduced in atopic dermatitis, which is shown here by these orange boxes at different skin sites during flares of AD. And the skin microbiome diversity has also been shown to inversely correlate with severity of the disease. And then in contrast to these pro-inflammatory effects that I've mentioned of Staph aureus, it's thought that the commensal skin microbiome, or the healthy species in our skin microbiome, may be beneficial and maintain homeostasis in the skin. So this includes the coagulase negative staphylococci, such as Staph epidermidis and Staph hominis, and it's thought that these may protect us against the pathogenic effects of Staph aureus. And similarly, other commensals such as Cutibacterium acnes and the gram-negative Roseomonas mucosa, they have been shown through in vitro and clinical studies to inhibit the growth of Staph aureus and actually even in some cases improve the symptoms of atopic dermatitis. We know that the early life skin microbiome initially depends on the mode of birth delivery. So babies born by vaginal delivery tend to have a skin microbiome which is more representative of the mother's vaginal microbiome compared to babies born by cesarean section who have a early skin microbiome, which is more similar to the mother's skin microbiome. And this has sort of led to questions around whether early disruption of the infant skin microbiome affects our future risk of inflammatory skin disease like atopic dermatitis. And there have been a number of birth cohort studies in which newborn skin microbiome is sampled, and then these babies are followed up and they're sampled at intervals in early life to see whether changes in the skin microbiome may be associated with future risk of developing atopic dermatitis. And this study, although it was small, found that there were changes in the skin microbiome at month 2, which showed that the microbiome was different in individuals who later on went on to develop atopic dermatitis at 12 months compared to those that didn't develop AD. So you can see these individuals on the left of this chart here who did go on to develop atopic dermatitis have a much lower abundance of this green species, Staph epidermidis, compared with those who were unaffected at month 12, who had increased Staph epidermidis at 2 months, suggesting that possibly Staph epidermidis is protective in the early skin microbiome for future development of AD. I mentioned that we have this strong association between Staph aureus and atopic dermatitis, and this figure summarizes some of the factors which underlie the enhanced Staph aureus colonization in AD. So a major factor is the dysfunction in the skin barrier in atopic dermatitis, but other factors such as reduced antimicrobial peptides in the disease and increased host proteases allowing the bacteria to penetrate into the deeper layers of the skin all are thought to make the environment more favorable for the growth of Staph aureus. And then Staph aureus itself produces a number of factors which are thought to drive inflammation and drive virulence in the disease. It produces a number of toxins which activate different components of the cutaneous immune system, and some of these go on to drive the key cytokines IL-4 and IL-13 in the disease, which again then go on to change the skin environment to make it more favorable for Staph aureus to grow. And then in contrast to these effects, the commensal bacteria are thought to be protective and maintain homeostasis. This is partly through competing for resources with pathogens like Staph aureus, but also through more indirect effects through producing phenol-soluble modulins and antibiotics, which prevent the growth of Staph aureus further. But we don't really understand how each of these many factors kind of works together in atopic dermatitis and which are the really important ones in determining these interactions between the skin microbiome and the host in AD. And so moving on to the virome and the mycobiome, or the fungal microbiome in atopic dermatitis. So we know that herpes simplex virus can cause eczema hepaticum, shown in these clinical photographs. And there has been some studies that show that there may be a link between Staph aureus infection and eczema hepaticum. So in vitro, this study showed that in the presence of Staph aureus alpha toxin, keratinocytes have a much higher HSV viral load compared to keratinocytes which aren't exposed to this toxin. So suggesting that potentially co-infection with Staph aureus plus herpes simplex that Staph aureus may actually predispose or increase susceptibility to infection with HSV. And in terms of the fungal microbiome, there really is very little data on this in atopic dermatitis. We know from healthy skin microbiome studies that the fungal microbiome diversity is reduced in adults compared to children where it's more diverse. But we need more studies looking at this in atopic dermatitis. So moving on to what we know about the skin microbiome in different geographical regions and in different ethnic groups. So there have been some small studies looking at this in healthy skin. And so this study showed kind of consistent differences between the skin microbiome in individuals from the United States compared with Tanzania. And then this study here looked at individuals from different parts of the US, as well as individuals from South America, and showed that based on the skin microbiome, individuals tend to cluster with others from their same geographical region. And this raises the questions around whether geographical or racial differences in the skin microbiome in atopic dermatitis underlie some of the different clinical phenotypes that we see of the disease. So we know that individuals with darker skin tend to have a more violaceous erythema, tend to have disease more on the extensor rather than the flexural surfaces, and tend to have more follicular AD phenotype compared to other skin types. And we don't really understand what's underlying this, and raises the question of whether these different individuals from these different regions have different microbiome signatures and whether these underlie some of these differences in the phenotypes. There has been one study which looked at Staph aureus strains from individuals with AD from different racial groups within the United States. And here they found that different groups had different subtypes or different strains of Staph aureus, and the profile, particularly of the superantigen genes, was different between these different groups. And these superantigens are thought to play an important role in how Staph aureus may drive inflammation in the disease. So this is another study suggesting that Staph aureus strain properties might be important. And then finally, I just wanted to mention a bit about the gut microbiome in atopic dermatitis. So we know that the gut microbiome is thought in early life is thought to be very important in the development of the immune system. So you can see on the left here that this the immune system goes under huge development in the first 3 years of life, and the early colonizing gut bacteria also evolves a lot over the first 3 years. And it's thought that these changes in the gut microbiome may be important in the maturation and development of the immune system. And this has led to a number of birth cohort studies in which the gut microbiome has been sampled at early life. And one of these was the EAT study, which was led by Carsten Floor from St. John's. And in this study, they followed up infants sampling the gut and the skin microbiome and looked at the effects of the early introduction of allergenic foods. And one of the interesting findings from this study was that infants with an increased abundance of this Clostridia species in the stool at 3 months were at more risk of developing atopic dermatitis, suggesting that possibly this gut microbe may have an inflammatory or immune modulating effect that has a role in the development of atopic dermatitis. And it's thought that changes in the gut microbiome may induce changes at distant sites, such as the skin, through what's known as the gut-skin axis. So this is where changes in the lumen of the gut, shown at the bottom here, induce changes in the epithelium of the gut and can make the gut lining more leaky. And this allows metabolites and microbes and stimuli to penetrate through the gut wall and enter the systemic circulation, and through that reach distant sites. And it's known that changes in the gut microbiome can have big effects on distant sites such as the brain, and it's thought to have implications in neurological and psychiatric diseases. And similarly, it's thought that these changes in the gut microbiome could induce changes at distant sites such as the skin. And so that's why there's a lot of interest, as well as the skin microbiome, but also in the gut microbiome in atopic dermatitis. And in terms of established AD, it has been shown that there are a number of changes in the gut microbiome in atopic dermatitis. Similar to the skin, there's reduced diversity in the gut microbiome, and there's thought to be increased pro-inflammatory species such as Faecalibacterium and Clostridium, and reduction in Bacteroidetes and Bifidobacterium. But we need more studies to kind of understand how these changes in the gut microbiome may mechanistically be kind of inducing changes in the immune system and in the skin. And so I think with that, I will just summarize that hopefully I've shown that new technologies have allowed us to really increase the way that we can investigate the skin microbiome through sequencing of microbial DNA. It's well known that the skin microbiome is associated with environmental factors such as skin site, and the skin microbiome is majorly altered in atopic dermatitis with increased Staph aureus, reduced commensal diversity. We know that early life skin microbiome changes might be important in disease development, and further work's needed to look into these differences in geographic regions and ethnic groups to see whether these groups harbour distinct microbiome signatures. And really, we need further studies to understand how these changes in the microbiome may interact with the immune system in the skin to potentially mediate diseases like atopic dermatitis. But it's certainly a growing area of research that has a lot of exciting potential for novel microbiome-based therapies for atopic dermatitis, whether these are topical therapies to modulate the skin microbiome or oral systemic therapies to modulate the gut microbiome, which I think Sandipan will talk more about. So thank you very much for listening, and very happy to take questions.